Welcome back to Cancer is a Mitochondrial Metabolic Disease Basics. Glutamine is cancer's second favorite fuel. Most people have heard that cancer cells love sugar, but most cancers also become highly dependent upon glutamine. And it's not just for energy, but for building blocks and antioxidant defenses. That's why glutamine targeting drugs like DON have been so interesting in cancer metabolism research. DON is a powerful glutamine inhibitor, but it also can be nearly impossible to access, can be difficult to tolerate, and not something that most people can simply just add to their metabolic therapy strategy. But here's where EGCG gets very interesting. EGCG, the main catechin in green tea, is not DON. It does not block glutamine metabolism in the exact same way, but studies suggest it may interfere with key parts of glutamine metabolism, including glutamate dehydrogenase, which inhibits the ability of cancer cells to ferment or make ATP from glutamine. So in this video, we're going to ask a very specific question. Can EGCG weaken cancer's glutamine metabolism? And given its accessibility and cost, could it be more useful than DON? We're about to find out. So without further ado, let's get into it. So today we are going to set out to talk about what effects EGCG has or doesn't have on the glutamine metabolic system used by cancer cells. And in the prior video, we focused on the left side of this classic C3 graphic looking at glucose metabolism. And on this particular video, we're going to be talking about the right side of the graphic, the metabolism of glutamine. And I'm going to start out with this paper here. We only showed what it wrote about glucose metabolism, titled The Roles of Epicatechin Gallate in the Tumor Microenvironment, Metabolic Reprogramming, and Immunotherapy. And it says here in the abstract, additionally, EGCG can suppress multiple metabolic reprogramming pathways, including glucose uptake, aerobic glycolysis. We talked about that in the last video. It also says later that glutamine metabolism, fatty acid metabolism, and nucleotide synthesis are also affected by EGCG. And that's where we're going to spend our time at today. So if you were to look at this graphic, we focused all of our time on glycolysis, and now we're going to look at the glutamine metabolism system on the bottom part of this graphic. It says in the paper, besides glucose, glutamine is another vital nutrient supporting tumor cell survival, as well as a crucial carbon and nitrogen reservoir necessary for tumor growth and proliferation, supplying ribose, non-essential amino acids, citrate, glycerol, essential for tumor metabolism, and supplementing the decreased oxidative phosphorylation caused by the Warburg effect. Evidence has demonstrated tumor cells can overexpress mitochondrial glutaminase, GLS, to catalyze the hydrolysis of glutamine to glutamate that is further converted to alpha-ketoglutarate by GDH or GLUD, glutamine dehydrogenase or aminotransferase. This means that the glutamine metabolic pathway rewired in TME, glutamine continuously replenishes the intermediate AKG or alpha-ketoglutarate for the TCA cycle to support the energy consumption caused by uncontrolled cell proliferation, leading to the consequences of cellular addiction to glutamine. The reprogramming limits the glutamine utilization by immune cells and regulates PD-L1 levels of cancer cells, thus affecting antitumor immune response and aiding tumor immune escape. One study found EGCG effectively reduces total and sodium-dependent glutamine uptake in tumor cells. Unfortunately, it is unclear from current studies whether this suppression is related to the expression levels of transport activities of glutamine transporters, including ASCT2 and SNAT1, SNAT2, although these glutamine transporters are often upregulated in tumors. Relevant evidence has revealed that transcriptional programs of these transporters are largely regulated by HIF1 alpha, ATF4, and MYC. Indeed, it has been observed that EGCG markedly reduced the expression of these transcription factors in tumors. Given that, we speculate EGCG may downregulate the expression levels of the above-mentioned glutamine receptors or transporters, but direct experimental results are still needed to confirm our hypothesis. In addition, to clarify the effect of EGCG on glutamine transporter activity, molecular docking simulations and dynamic analysis should be performed to explore the direct interaction of EGCG with them. So basically what these authors are saying is that EGCG likely has diverse effects on cancer's glutamine metabolism. And they then start to hypothesize, well, could it be through these different transporters? And I didn't know the ACT and SNAT1, SNAT2 transporters, what they actually were in terms of their SLC numbers. But I created this table at the bottom to actually orient us, because a lot of times in graphics and papers, they mention SLC1A5 or 38A1 or 38A2 more commonly than some of these other names, which is kind of funny because this is called the common name. But that being said, they're basically saying we don't know if it's these transporters. We don't know if it's only because of HIF1 and MYC signaling, which is kind of like largely responsible for metabolic reprogramming. And they also hypothesize that potentially if they did some docking simulations, they could even find out whether or not this is just expression-based downregulation or if this is actually directly blocking these transporters. So they're just still kind of scratching the surface of what EGCG can do when it comes to glutamine metabolism in terms of uptake. Where it's more clear is on this glutamine dehydrogenase, which we're going to spend a great deal of time on in this video. It later says in tumor cells, GDH or glutamine dehydrogenase is the metabolic bridge between glutamate derived from glutamine metabolism and the TCA cycle. Similar to targeting rate-limiting enzymes in glycolysis, targeting GDH also can interfere with the energy generation of tumor cells. Therefore, there have been an increasing number of studies, including tumor metabolomics analyses, shedding light on the inhibitory roles of EGCG in tumor glutamine metabolism by targeting GDH. Previous investigation demonstrated EGCG reduced GDH expression to disrupt glutaminolysis in primary effusion lymphoma cells, and EGCG-induced tumor cell death was attenuated by if the deficient alpha-ketoglutarate was supplemented. Meanwhile, a scientific study suggested EGCG could dose-dependently target GDH activity, which leads to increased oxidative stress and thus makes tumor cells more sensitive to radiotherapy. Corresponding molecular dynamics simulations supported the abilities of EGCG to establish multiple non-bonding contexts at the adenosine diphosphate ADP activation site of GDH and to form multiple hydrogen bonds with a variety of amino acid residues of GDH. These results suggest that EGCG can block glutaminolysis for energy supply of tumor cells. And this is critical. So basically what these authors are saying is that there is evidence to suggest that EGCG is downregulating glutamine dehydrogenase expression. Less protein is available to convert glutamine into glutamate. But also there's evidence to suggest that EGCG can directly inhibit by binding to GLUD or GDH or glutamine dehydrogenase as a secondary mechanism. And that's really the big piece of what we're going to be exploring today. So the next paper I'm going to pull is titled Targeting Glutaminolysis to Treat Multiple Myeloma, an in vitro evaluation of glutaminase inhibitors telaglenastat and EGCG or epigallocatechin 3 gallate. It was published in 2023. It says here at the bottom, telaglenastat and EGCG act synergistically to inhibit cell proliferation and promote apoptosis in KM3-BTZ cells, possibly by targeting glutamine metabolism and glycolysis. And again, this is a study looking at multiple myeloma. And so I pulled this graphic out of a paper to prove a very important point. It's very likely that cancer cells are using glucose and glutamine at varying ratios depending on nutrient availability and the need of that particular cell at that particular time. So as we see here, these are hepatocellular carcinoma or liver cancer cells. And what they're doing is they're depriving these liver cancer cell types of glucose. And they're looking at what level of consumption is happening of glutamine at 0 hour, 24, and 48 hours respectively. So we see here that the baseline amount of glutamine utilization by these two different hepatocellular carcinoma or liver cancer cell lines is right here at this white bar here. And then at low glucose, hour 24, you see how at 24 hours, both will rise significantly. This particular cell line rose about 2.5 fold, and this cell line rose about 4.4 to 4.6 fold. And then what's even more dramatic is at 48 hours, you have a 6.2 to 6.4 fold of this particular cell line, and you have a 5.1 to 5.3 fold increase in this particular cell line. So when you were to deprive these cancer cells of glucose, they just keep chugging along if glutamine is in the environment, which is why a ketogenic diet or blocking glucose metabolism is not enough for most people. Now, that dovetails nicely into this paper that was published in January of 2022 titled Glutamate Dehydrogenase 1 Mediated Glutaminolysis Sustains Hepatocellular Carcinoma Cells Survival Under Glucose deprivation. So this experiment was interesting because what they did was they looked at, okay, what happens to these different hepatocellular carcinoma cell lines when we give varying amounts of glucose? So they did. They gave 0.1 ultra low, 0.5 ultra low, 1 millimole, still very low, 2 millimoles, and then very high. In fact, 25 millimoles is equivalent to 450 milligrams per deciliter in the bloodstream. So huge amounts of glucose. And you see here that at ultra low glucose, you see very little cell growth. And then as you give varying amounts more of glucose, then you see more cell growth. What I thought was fairly interesting is that there's not a huge amount of difference between 1 millimole and 25 millimoles. So you see here at the end of these 4 days, you have about, if I had to estimate with my eye here, I'd say it's like maybe like 42 or 43. And then at the end of 4 days, if you give them much more glucose, 25-fold more glucose, you see about 60. So there's definitely more growth, but still, it's not like low glucose alone stopped these cells. And also, no one's going to be at 1 millimole of glucose in their bloodstream, and no one's definitely going to be at 0.5 or 0.1 millimole of glucose in their bloodstream, right? That's impossible for anybody to achieve. So what they did was they looked at EGCG on these hepatocellular carcinoma cells at either high glucose or low glucose and at varying concentrations of EGCG. So let's start with the high glucose. So if they gave 0, 25, 50, and 100 micromoles of EGCG, you see that there is some small amount of growth inhibition at the end of 4 days if you give higher amounts of EGCG. Actually, if you actually look at the endpoint here, they kind of look like 25, 50, and 100 kind of end at the same place, even though earlier on 100 micromoles seems to have a better outcome here. But at 0, it's clearly the highest, right? So if they got high glucose and no EGCG, it showed the highest growth. But this is where it gets fascinating. So look what happens when they put it in a low glucose medium and they put in EGCG. So this is what happened when there was no EGCG, right? At a low glucose medium, all the way from 1 to 25, you saw basically exponential growth, maybe slightly less growth here at 1 millimole compared to 25. But when you started adding EGCG, here's 0, right? Here's basically equivalent to this line here. But when you started adding EGCG at 25 millimole, you saw a huge decrease. And then at 50 and 100 micromoles, you see almost all the cells are gone at that point. So this is the importance of targeting both glucose and glutamine simultaneously, not one at a time, both simultaneously, because both can be relied upon by these cancer cells. And as it said here in this prior graphic, it can upregulate the needed machinery and metabolism capacity of glutamine pretty quickly in high fashion, as you see here on this graphic. So we have at this point talked about how EGCG has likely effects at the level of SLC1A5, SLC38A1, SLC38A2, GLUD, both in expression and likely direct blockade. We have not talked about this yet, but we will in future videos. But this is also part of the glutamine system. But that still doesn't answer the question, is EGCG better than DON? So this is just a graphic to get us oriented here. So here's SLC1A5, here's SLC38A1, A2 would be like a slightly different version of that. Here's 7A11. Here is GLUD, or as this publication calls it, GDH. Here's GDH or GLUD, glutamate dehydrogenase, converts glutamate into alpha-ketoglutarate. Here's where DON blocks, GLS, DON, right here, from glutamine to glutamate. So they are mechanistically different, but they are both intimately involved in the metabolism of glutamine and the utilization of glutamine by cancer cells. So let's take a look at this paper that was published in April of 2006, oldie but goodie here. Green tea polyphenols modulate insulin secretion by inhibiting glutamate dehydrogenase. And I'm going to skip to the graphic here. So what they did was they looked at the amount of activity of glutamate dehydrogenase at varying concentrations of different green tea polyphenols. And here's the different polyphenols they use: epicatechin, epigallocatechin, epicatechin gallate, epigallocatechin gallate, EGCG, right? Here's EC, here's EGC, here's ECG, and EGCG. And you see here that basically these two catechins here are, I don't want to say useless because there may be other benefits that it has on the cell, but they're effectively, for this particular enzyme system, effectively useless. Now, if we look at the EGC and EGCG, we see a much different story. As a matter of fact, we see an exaggerated response at as little as 1 micromole of EGCG, and we see a slightly more dose response all the way up to 10 micromoles for this particular enzyme. And this is a bovine, so it's a cow enzyme. And we'll see how that stacks up compared to human enzyme or other animal enzymes. So this is a study that was done where they looked at head-to-head DON and EGCG. And what they did is they looked at the baseline amount of glutamine metabolism with 0 EGCG. And then they looked at the amount of decrease at increasing amounts of EGCG. And you see here that there's a dose response. So at 5 micromoles, we see that it goes down from like 40 to 30. And then at 10 micromoles, it's down to about 25. And about 20 micromoles, it's like maybe 20, I don't know, 22, 23. And then it kind of levels out right around 30 to 50 micromoles. And then what they did later, which was pretty cool in this study, they head-to-head looked at DON and EGCG. So here is if glutamine is added and no inhibitors are added, right? Here's kind of like if they let the cancer cells go, right? And then here's 20 micromoles. So here's right here on this graphic here, 20 micromoles of EGCG. We see a huge decrease in glutamine metabolism here. And then they added 40 micromoles of DON. And what we see here is we see a very comparable decrease in glutamine metabolism. Yes, they work in different ways. Glutaminase is a different enzyme than glutamine dehydrogenase, but they are in the same line of metabolic processes, and that would inhibit alpha-ketoglutarate from getting into the TCA cycle so it can be metabolized through mitochondrial substrate level phosphorylation or the fermentation metabolism of glutamine. So it is blocking at different levels this process and ultimately having very similar effects in this particular study. Matter of fact, I had AI basically better than me than I could do guesstimate the percent decrease of glutamine utilization by these cell lines at varying concentrations. So at 5 micromoles, there was about a 25 to 30% decrease. At 10 micromoles, there was about a 32 to 36% decrease. And at 20 micromoles, there was about a 40 to 45% decrease. I think it's important to memorize or at least have in your mind these particular concentrations, because I'm going to pull back to this particular paper that was published in March of 2024. We went over this in the last video, but I pulled out the snippet about EGCG bioavailability and obstacles. And this is basically a continual problem for whether it be drugs or nutraceuticals. EGCG bioavailability obstacles. Oral absorption of EGCG was noticed to be very low. When EGCG was given as oral administration in rats, its absolute bioavailability was 0.1%. Some in vitro studies stated that the active concentration of EGCG was about 1 to 100 micromoles per liter. Unfortunately, the maximum EGCG concentration in plasma were reported to be in the lower values, 0.16 of ingested catechins. This can be explained as EGCG is known for its minimal stability and low bioavailability. This is where there's kind of a point of contention. So yes, for getting into the bloodstream, very important, especially when we're trying to compare, whether or not we can try to approach these concentrations in the bloodstream. But a lot of the magic that happens with these flavonoids and these nutraceuticals and plant compounds is that they don't get absorbed. They block brush border enzymes and lower glucose uptake into the small intestine, or they interact with the intestinal microbiome and they have a kind of a backdoor effect through the colonic microbiome and certain compounds that are created by those gut bacteria. But again, specifically looking at blood concentrations and trying to approach these micromolar quantities, there are obstacles that need to be overtaken. One of the main reasons behind the low bioavailability of EGCG is intestinal metabolism. Intestinal metabolism affects the bioavailability of flavonoids. It has been shown that EGCG is easily metabolized by Rhizotella plenticola, Enterobacter aerogenes, Klebsiella plenticola, Bifidobacterium longum, and Klebsiella pneumoniae. These types of bacteria are found frequently in gut microbiota. Using a pig cecum model, it was proved that the ester bonds of the gallate derivatives were not durable versus microbial digestion. By using the rat model, after oral consumption of 25 milligrams of EGCG, the fecal metabolites resulted in 9 metabolites detected in feces. Nevertheless, no EGCG was detected. In addition, there were studies supporting that EGCG may be even degraded and countered by metabolism even before reaching the colon, especially in the small intestine. Therefore, there is an urgent and continuous need for improving EGCG bioavailability. One of the practices that is utilized to increase EGCG bioavailability is using other natural products such as curcumin or piperine. Curcumin was reported to inhibit P-glycoprotein pump, thus inhibiting EGCG efflux, while with piperine, co-administration of EGCG and piperine enhanced the area under the curve and maximum plasma concentrations by 1.3-fold related to mice treated with EGCG alone. Furthermore, nanotechnology has improved EGCG bioavailability. EGCG has been prepared using different nanotechnologies such as nanoemulsion, nanoparticle, and nanoliposome. All of these technologies have been shown to increase EGCG bioavailability by different mechanisms. So I use a couple different AI models to see what the literature has for us as a way to estimate serum concentrations by varying degrees of oral EGCG, whether it be a standard formulation or whether it be a liposomal formulation. And we see here that at 400 milligrams, we're at about 1.24 micromoles. At 800, you're about 2.10. 1200, you're at 4.02. 1600, you're at 6.35. And at 2000, you're at 7.5 to 8.5, which would be quite a bit higher than the daily recommended dosing. So these are probably doses you'd probably want to avoid. That being said, I've heard many anecdotal stories of people taking much more than the recommended amount and having really no problems whatsoever. Consult your doctor before approaching more than the recommended amount, of course. Now, let's turn to liposomal. Liposomal absorbs quite a bit better. So at 400 milligrams, it's estimated to be about 3.8 micromoles. At 800, 6.52. 1200, 12.45, and at 1600 milligrams, it'd be nearly 20 micromoles, and then above 1600 would be higher in the 23 to 26 micromolar range. I had Gemini do the same calculations. It came to similar conclusions. Basically, probably very little chance, kind of normal dosing of EGCG to approach the concentrations we would want. However, I do think that given the concentrations we've seen, for example, let's say the gold standard is somewhere between 10 to 20, probably 20 being the realistic gold standard for what we're trying to look for at inhibiting glutamate dehydrogenase and glutamine metabolism, being 20 comparable to DON. That seems to be fairly achievable with liposomal at either 1600 to 2000 milligrams. Again, that would be higher than most publications recommend would be used. That being said, again, these are things you have to talk about with your doctor, track your liver-related enzymes, and figure out if you're able to tolerate it. But if you are, that would have probably substantial amount of glutamine blockade at the level of GLUD. Remember, at 5 micromoles, you're about 25 to 30% decrease estimated. At 10 micromoles, you're about 32 to 36% decrease estimated. At 20 micromoles, you're about 40 to 45% decrease estimated, which was in this paper very significant and very similar to what DON was able to do at 40 micromoles. So this is the drug, this is the EGCG. The dose and formulation are very important with these nutraceuticals and drugs as well. Unfortunately, there is no published data on EGCG for outcomes. And the outcomes that I really care about are recurrence and does it decrease your risk of death? But what we do have is we do have this paper that was published in October of 2024 and it was titled Pre-Diagnosis Tea and Coffee Consumption and Survival After a Diagnosis of Ovarian Cancer Results from the Ovarian Cancer Society Consortium. And what it says here is that compared to women who did not drink green tea, any green tea, consumption of 1 or more cups per day was associated with a better overall survival, a hazard ratio of about 0.84, which is a 16% reduction in death if you drank 1 glass of green tea or more per day. A similar association was seen in ovarian cancer-specific survival in 5 studies with this information at a nearly 20% reduction in death if these women drank more than 1 cup of tea per day. Is this equivalent to EGCG? No, it's not. This is different. This is an entire plant compound, whereas EGC is a very specific extract of this plant compound. That being said, I'm still very bullish on EGCG because not only do I think we can potentially achieve these type of concentrations with liposomal or later on, maybe like nano liposomal or micellar formulations pretty easily, and EGCG is readily available. It's relatively cheap and much easier to find compared to its big brother DON. So if you like videos like this, please like share, and subscribe. And until next time.